And then we also learn about the reabsorption, all right, and also secretion at the distal convoluted tubule. But this one is under the control of hormone also. So what is the name of the hormone involved here, Myra?
ADH and aldosterone.
All right, very good. All right, aldosterone and also ADH. Okay. Okay, so let's continue. Right, kita ada double period today. Kalau kita habis awal, cikgu beri masa for you to do your own revision lah as a preparation for your test, right, this afternoon. Tapi cikgu nak jumpa Arif kot for AI consultation. Tapi kan Arif ada kat sini kan. Sepatutnya boleh apa face to face lah. Tapi tak apa. Kita habiskan dulu syllabus. After that cikgu confirm lah what to do. Alright. So we continue today. Alright. So we have covered until alright, the collecting duct. Okay. Alright. So yesterday we learned about ADH controls the absorption of water in the collecting duct. Alright, and then we learned that when there is an increase in osmotic pressure, blood osmotic pressure, alright, so meaning that there's more water, right, a lot of water, so therefore we need to remove more water from the blood. Betul? Alright, so this will be detected by the osmoreceptor cells in the hypothalamus, and then next sending a message, right, to the pituitary gland. Okay, and then reducing the release of ADH, causing the collecting duct to be less permeable to water. All right, less permeable. So that less water will be reabsorbed. So more water will be actually excreted with urine. So that we will produce diluted high volume and diluted urine. Okay, and then if let's say blood osmotic pressure is low, meaning that less water in the blood, we need to absorb more, right, of the water from the urine. Okay, so this will cause pituitary gland to secrete more ADH. All right, and then secretion of ADH will increase permeability of the collecting duct. All right, to water, and an increase in permeability, as I said yesterday, is due to the presence of aquaporins. Remember aquaporins? So what are aquaporins? Aquaporins are water channel that allow for water to pass through the membrane. So actually last time we learned about the phospholipid membrane, right, phospholipid bilayer, water can actually pass through, right, in between of the phospholipid molecule. However, with the presence of aquaporins, this will increase, right, the amount of water which can actually pass through the membrane. So this is how, right, the permeability of the collecting duct is controlled. Alright, macam ni kita boleh kurang atau tambahkan the permeability of the collecting duct with or without the presence of aquaporins. Alright, and this aquaporin actually is stimulated, right, by the presence of ADH. Maksudnya kalau ada ADH banyak release, then aquaporins will be greater in number, alright, increasing the permeability. When ADH is withdrawn, less ADH, meaning that this will reduce the number of aquaporin, alright, so that the membrane become less permeable to water. Okay, so now today we're going to look into aquaporin. Okay, so this is, right, aquaporin. So as you can see here, so this is the, look at the picture given here. Right, so this is the extracellular fluid, all right, and this is the plasma membrane, the phospholipid bilayer, all right, and this is aquaporin channels in a vesicle. ni aquaporin ni. Aquaporin ni this protein channel, right? Protein channel that allow for movement of water passing through it. All right. So so about this ADH, right? ADH stimulation of aquaporin channels. So ADH stimulate, right, more aquaporins to be present on the plasma membrane of the collecting duct. Right, so the osmotic gradient created by the counter current multiplier system provide the force for water reabsorption through the collecting ducts. So this one is depending on osmotic potential. All right. So the rate of osmosis across the walls of the collecting ducts, right, or the permeability of water. Permeability of water, so meaning that this one, right, the rate of osmosis is depending on the permeability, right, of the wall to water. So the number of aquaporins. So aquaporins are water channels in the plasma membranes of the collecting duct epithelial cell. So meaning that this one, right, the permeability is determined by the number of aquaporins or water channels in the plasma membrane of the collecting duct epithelial cell. Okay, all right. So when ADH is absent, right, when ADH is absent, aquaporin channels are located in the membrane of intracellular vesicles within collecting duct epithelial cells. So you can see this one is without ADH. When there is no ADH, aquaporins are located in the vesicles within the cell, in the cytoplasm of the cell. So you can see, right, this vesicle containing aquaporins here. Okay, so dia duduk di mana? Dia duduk di dalam sel, cytoplasm of epithelial cell. All right, and then when ADH is present, all right, so with ADH stimulates the fusion of these vesicles with the plasma membrane. So when ADH is present, what happens is these vesicles, right, will be stimulated to move towards the plasma membrane and fuse with the membrane. Okay, this remind me of what process? Remind you of what process? What process is this? When this vesicle from the inside, from cytoplasm moves. Exocytosis, very good. All right, exocytosis. So these vesicles then move toward the plasma membrane and fuse with the membrane here, the plasma membrane. Right, therefore, adding these aquaporins to the plasma membrane. So you can see here. Right, it fuses with the plasma membrane, so that adding the aquaporins, right, to the plasma membrane. Okay, alright, so nampak tak ni dia cantum sini, oh sinilah, patutnya sini so dia cantum fuses, alright, and it becomes like this. Okay, so now there will be more aquaporins present on the plasma membrane. Alright, with the presence of ADH, therefore, this will increase the permeability of the collecting duct for water. So more water will be reabsorbed. Okay, alright, and then when ADH is withdrawn, right, so sekarang blood osmotic pressure goes back to normal, right, we no longer need to reabsorb more water, so decrease the secretion of ADH, so ADH is withdrawn, so without ADH, all right, what happened next is the plasma membrane, this one, the plasma membrane pinches inward. So this remind you of what process?
Endocytosis.
Endocytosis, very good. All right, endocytosis. So pinches inward, right, in endocytosis to again form an intracellular vesicle. So form, right, a vesicle inside of the cell again. So it goes back to this one lah. All right, so it becomes like this again, right? So aquaporin stored in the vesicles inside of the cell, in the cytoplasm of the cell, removing aquaporin channels, right, water channels from the plasma membrane, therefore decreasing the permeability of the plasma membrane towards water. All right, so that less water will be reabsorbed when ADH is absent. Is that clear?
Yes,
Faham ke?
teacher.
Yeah, teacher.
Alright, very good. Do you have any question? Ada soalan tak?
No, teacher.
Ada. Alright. This one is an additional lah, cikgu tambah, so that when aquaporin is mentioned, you know what it is. Alright, so it is actually referring to water channel. Okay. Alright, this is more diagram showing. Alright, aquaporins, aquaporin also here. So this is blood and this is lumen of the collecting duct tubule. Alright, so nampak So aquaporins spore are inserted into the cell membrane, increasing flow of water out of tubule. Alright, so sekarang dia pergi jadi aquaporin. Okay, increasing number of aquaporin. So same lah, this one I put a lot, right? Later you study on your own lah. Tengok you read on your own. Sama saja explanation. Alright, so now we are done with everything, that all of the processes, right, that occur in the nephron, in the kidney. Right, we have discussed starting from that blood that goes in, blood that goes in carried by renal artery into the kidney. Alright, and then after that, this artery form many smaller branches, the arteriole, and the blood vessel that carries unfiltered blood, alright, is afferent arteriole. Alright, so carries the blood into the glomerulus. Okay, and then the blood goes out through efferent arteriole, which is smaller in diameter, creating high pressure in the glomerulus, causing, right, the small molecules to be filtered out from the blood. Okay, all right, and this is the ultrafiltration process. Okay, ni kita wrap up, kita wrap up the whole processes. So since kita wrap up, cikgu tanyalah. Alright. So the ultrafiltration process, alright, these small molecules we need to pass through several layers. Alright. So apa dia yang these small molecules kena pass through ni? Alia?
Ultrafiltration kan?
Yeah, the ultrafiltration process, where small molecules are forced out due to high pressure generated inside of the glomerulus. Right, but so these small molecules are filtered out through, right, ada several layers. So apa dia layer yang jadi filter ni?
Glomerulus. Yang tu ke? Macam tu ke soalan teacher?
Tak ada soalan cikgu, apakah lapisan-lapisan yang perlu dilalui oleh these small molecules through, right, before becoming the glomerular filtrate?
Basement membrane.
Right, very good.
Podocyte.
Okay, very good.
Fenestrated capillaries.
Ya, dah. Lagi?
Tak, teacher.
Yes. All right, very good. So, fenestrated wall of the capillary, all right, and then followed by the basement membrane, all right, and finally the podocytes. Okay, and then becomes glomerular filtrate. Okay, and then next part is what is the process that is happening after that? Fakrul?
After ultrafiltration,
Yeah.
Selective reabsorption.
Selective reabsorption process. All right, okay, very good. So next is selective reabsorption process, because we know that during the ultrafiltration process, all right, all small molecules will be filtered out, including the useful substances. Betul, glucose, amino acid, mineral ions, all right, so all of these are useful substances, including water. So therefore, right, we need to reabsorb all of these things back into our blood. All right, so next process is selective reabsorption process that occurs at the proximal convoluted tubule. Will reabsorb, right, all of these useful substances back into the blood. Okay, so example yang itulah. Mechanism cikgu tak tanya because nanti takes time lah pula. Okay, so glucose, amino acids, alright, water and ions also. Okay, next, loop of Henle. Alright, so next, right, the filtrate then goes further to the loop of Henle, going down, right, the descending limb of the loop of Henle. So what actually happens here, as Hazim,
Yes, teacher. Down the loop of Henle,
yeah, descending limb of the loop of Henle.
descending loop. Okay. The membrane become permeable to water and impermeable to Na plus ions.
Okay, so what happened?
Um, solid concentration increases in the medulla because
Apa pula Azim? You tanya what happened
sorry.
at the descending
What
limb of
happened?
the loop of Henle.
What? Teacher nak yang mana satu? Teacher? What
Descending
happened?
limb. Descending limb property just now, right? Ada betul dah. So descending
Ah,
limb is
what
permeable
water?
to water but not to salts. Alright, so as the filtrate moves down the descending limb, what is going to happen?
Water leave the filtrate.
Alright, very good. So water leaves the filtrate by osmosis.
Osmosis.
Alright, and then filtrate becoming more
Solid.
more diluted,
More
more
ada
concentrated.
solid boleh teacher kalau
Filtrate.
more
Dia glomerular filtrate tadi tu. Dia lah lose water kan? More concentrated. Alright. So descending limb of the loop of Henle, it is permeable to water but not to salts. So as the filtrate moves down, descending limb, alright, so it loses water. Okay, but then salts trapped inside. So the filtrate will become more concentrated as moving down the descending limb of the loop of Henle. Alright, okay, next. What happened? Letitia?
And then dengar ke teacher? Sebab
Dengar,
my mic rosak. And then the ascending limb, first Na plus will diffuse out through facilitated diffusion because filtrate is more concentrated with Na plus. And then after that, more Na plus will leave the filtrate by active transport because as the loop of Henle ascends, sodium concentration will decrease. Sebab dia keluar. Yeah, that's
Alright, so that's the reason why it will be pumped out, right, by active transport. And then the pumping process, right, using energy, the objective is to increase the solid concentration in the medulla. Betul? Right, because it's very important to make sure that medulla has high solute concentration or high osmolarity to allow for water reabsorption. Betul? Okay, okay, very good, Letitia. Yes. Okay, ingat Arif nak tanya soalan. Alright, okay, so we know that at this ascending limb of the loop of Henle, it is permeable to salts but impermeable to water. Alright, so as the filtrate ascends the ascending limb at the lower, at the bottom region, right, known as the thin segment, and right, or the sodium ions actually leave by passive transport. Alright, because due to their high concentration in the filtrate compared to in the medulla, and then as it ascends further, alright, further up at the thick segment, sodium ion will be pumped out by active transport into the medulla. Alright, so here energy is needed, and energy is actually spent, right, to make sure that the medulla has high solute concentration to allow for water reabsorption right, to happen at the descending limb of the loop of Henle as well as at the collecting duct. Okay, all right, and then at distal, what happened next? Afika.
Yes, teacher. At distal, it will have reabsorption and secretion, which is the reabsorption of water, but it depends on the salt and water concentration. So at this process, we have hormone ADH that helps to control the reabsorption. So when
ADH helps reabsorption of water. All right, and then another hormone involves?
Aldosterone. Aldosterone. Yes, aldosterone.
All right. Okay, very good, Afika. All right, very good. Okay, next ada collecting duct. Okay, collecting duct kita dah discuss tadi kan. Cuba tak tanya dah lah. Okay, okay, very good. Alhamdulillah. Okay, so kita dah wrap up all of the processes, right, that actually happen along the nephron in the kidney. Okay, sudah habis dah that part. So now we continue with sikit lagi ya. So next. Okay, this is about relating or correlating, right, the length of the loop of Henle to the need for water conservation in animals. All right, so last time we learned that loop of Henle, all right, loop of Henle, the main, right, role of the loop of Henle is to generate high osmolarity in the medulla. Alright, so sebenarnya the longer the loop of Henle is, right, the more concentrated, right, the medulla becomes. Sebab dia akan banyak, because the filtrate will be very concentrated and it will like pump out more salts, right, or sodium ion into the medulla. So lagi panjang, lagi pekat. Okay. So now the length of the loop of Henle is positively correlated with the need for water conservation in animals. So if let's say animals that live in the desert, for example, all right, so they need to conserve a lot of water. Maksudnya, right, dia akan ada longer loop of Henle. Because they, by having like longer loop of Henle, they can be absorb more water. Okay. All right. So for example, here the kangaroo's rats, all right, loop of Henle is much longer than that of other rodents. So this in part explains the kangaroo rat's amazing ability to survive in deserts. All right. So water is scarce, then they need to conserve more water, and then their adaptation is by having long loop of Henle, right? Longer loop of Henle so that they can conserve more water, because more water can be reabsorbed. All right, so the kangaroo's rat's kidneys are especially efficient in, and produce only small quantities of highly concentrated urine. Okay, sebab dia nak conserve more water tadi kan, so they will reabsorb a lot of water so that the urine produced, right, will be in small quantity and highly concentrated. All right, so they have very long loops of Henle, which builds a higher ion concentration in the medulla. Faham tak? Can you relate this? The longer the loop of Henle is, the more ions can be pumped out from them, right? Creating higher osmolarity or solute concentration in the medulla. All right. So this therefore allowing more water to be reabsorbed in the collecting duct. Faham tak? Sebabnya lagi panjang dia, lagi tinggi concentration ions dia. Alright, so that as the urine, right, passes down the collecting duct, and then the outside too always, right, is hypertonic. So that water keeps on leaving the urine from the collecting duct and then back into the blood, so that more water can be reabsorbed. All right. So kangaroo rats can concentrate urea to 3,500 milliosmol per liter, whereas compared to humans can only concentrate urea to 400 milliosmol per liter. Alright, maksudnya dia lagi greater lah, lagi efficient in conserving water. Faham? And this is very much needed, right, in their habitat. Tapi kan, lepas ni you all boleh cari kot berapa panjang dia sebenarnya. Right, this loop of Henle ni. How long it is. Okay, tapi janganlah compare dengan kita punya. So kita punya sel besar compared to dia orang punya sel ni mungkin kecil. Compare dengan yang setara dengannya. Alright, other rodents here. Okay, this is more, this is additional lah. Alright, so the ion concentration in the medulla bales, right? The ion concentration in the medulla bales as the loop of Henle descends. Alright, so a longer loop of Henle in animals implies a larger medulla. Okay, compared to the kidney size. So loop of Henle kan duduk dalam medulla. Alright, most part right duduk dalam medulla. So that if let's say the animals they have longer loop of Henle, right, we can like make assumption that they have the medulla right, of the kidney is larger. Right, to the size of other parts lah. Macam cortex dia smaller lah kot. Dia medulla lagi besar. Okay. Then in animals with a shorter loop of Henle, right? So it shows the relationship between relatively medullary area in the mammalian kidney and the maximal urine concentration that can be produced. So you can see small size of medulla, small medulla and large medulla. So large medulla implies that the animal, they have a longer loop of Henle. And then look at this one, the y-axis is, wait, cikgu besarkan. Y-axis shows maximal urine osmotic concentration, right? The osmotic pressure, showing the water lah, right? Water level. Okay, so nampak tak the medullary, larger medulla has higher, right? Urine osmotic concentration. Alright. Tapi ni IB tak tanya kot . Okay, this example of question, explain osmoregulation in the kidney. Tapi ni soalan lama tau. Nanti you all carilah soalan baru. But then the points are about the same, I guess. Alright, so first is osmoregulation. First is the introduction, right? Osmoregulation takes place in the loop of Henle and collecting duct in the medulla. Alright, and then you talk about loop of Henle. After that, you talk about collecting duct. Alright, so boleh dapat lah 8 markah ni kan. If let's say you explain what is happening at the loop of Henle, what is happening at the collecting duct, you can easily get 8 marks. So descending limb, permeable to water but not to salts. Descending limb, permeable. Ada salah tak sini? Ada mistake tak? Is there any mistake?
Ada.
Yes, ni kan? This one patutnya apa ni?
Permeable to sodium.
Ascending.
Ascending limb. They should be ascending limb is permeable to sodium ion but not to water. Alright. Okay, and then sodium ion is pumped out at the ascending limb into medulla, generating osmotic potential between nephron is tubule, and medulla, some water leaves. Descending loop by osmosis. Alright, output is reduced, reduce salts concentration. Alright, and then collecting duct here, filtrate enters collecting duct from distal tubule, counter current flow of blood in capillaries and filtrate in duct. Okay, this one is the flow, counter current flow of the filtrate in the tubule and also blood in the capillaries. So maintains concentration gradient, alright, or osmosis of water into blood. And then dehydration, this one dia cakap pasal osmoregulation ni. Dehydration detected by hypothalamus leads to the release of ADH by the pituitary gland. ADH opens aquaporins, outer channels in walls of duct, increase transfer of water into blood, therefore producing hypertonic urine, small volume concentrated urine. Excess water in blood leads to breakdown of aquaporins and producing hypotonic urine. Okay, tapi kan, because sometimes we do not know, right, what are the points in analysis scheme. Kalau cikgu, don't simplify this way lah, excess water, right. Cerita balik, excess water results in detected, right, again, repeat this one again, detected by hypothalamus and then lead to reduction, right, of the release of ADH, decrease in the release of ADH, and cerita balik semua ni, semula. Right, to be safe. Jangan don't simplify it this way. Ni bahaya ni. Faham? I think this is more recent punya question. Explain how the structure of the nephron and its associated blood vessels enable the kidney to carry out its function. Alright, but this one kena cerita pada awal lah ni. Alright. And enable kidney to carry out its function. So you talk about osmoregulation or excretion, starting from the ultrafiltration. See, sebab ni dia tak spesifikkan di mana. Alright, we have to start. We have to like explain everything until collecting that. Okay, so done with that. Alright, so next is U2. Banyak lagi kan, U2, A1. Itu. Itu je. 3 more. Alright, 1 understanding, 2 applications. Alright, so this is next understanding. Alright, U2, the Malpighian tibial system. Right, in insects and the kidney carry out osmoregulation and removal of nitrogenous waste. So we have learned about the kidney, right? We learned that kidney carry out osmoregulation and removal of nitrogenous waste. Okay, so in insects, right, they have this system known as the Malpighian tibial system. Okay, which will carry out osmoregulation and removal of nitrogenous waste. So kalau kita ada kidney, insects dia ada apa? Dia ada Malpighian tibial system. Alright, this is example of grasshopper and insect. Alright, so this one is the introduction lah. So first is you must know what is hemolymph. Alright, so sebenarnya there is a detailed explanation dalam IB guide. Cikgu dah ambil gambar letak kat sini. Alright, nanti you can refer to your, kan cikgu dah share kan? Buku tu kan?
Dah.
Kau pernah tak ke buku-buku yang apa, soft copy yang cikgu dah share sebelum ni? Have
Ada,
you
teacher.
referred to this reference book? Tak
Ini,
pernah
teacher.
ya? Apa dah? The IB study guide yang masuk Oxford tau.
Teacher ada bagi ni kan, the
Oxford yang masuk IB study guide. Ada 2 kan?
Textbook teacher.
Alah, awal semester dulu kan. Cikgu pun tak ingat dah. Tapi I have shared everything. You all.
Ada teacher kat email.
Dekat mana?
Email.
Nah, cikgu tak ingat lah dulu macam ni you all ambil. Tapi I have shared with you. Early SEM 1 dulu. Alright. You all pernah refer ke buku-buku ni? Ke simpan dekat email tak buka-buka?
Refer.
Refer, teacher.
Right, because this IB study guide is very useful, you know. Right? Kalau nak beli harga dia 100 lebih. Tapi you all dah ada e-book. Cikgu dah share e-book dan you all tu. So tak payah beli pun. Baca je kat situ. Right? This one I took it from IB guide. IB study guide. Right. So, so this is the Malpighian tibial system in insects. Alright. So first, right, you must know what is hemolymph. So hemolymph is referring to a fluid that is analogous to the blood. So like us, kita ada blood. So kalau insect, they call it as hemolymph. Right, that circulates in the interior of the insect's body, remaining in contact with the tissues. Okay, and this semua hemolymph ni hemolymph outside ni. So in contact with the other tissues here. So hemolymph similar to our blood. Maksudnya dia adalah semua right, components. And then next is the Malpighian tubule system, the Malpighian tubule. So this green color structure is actually the Malpighian tubule. So ni tubule dia lah. Alright, so Malpighian tubules are longer and more convoluted than shown in this simplified illustration. So yang shown here ni very simple structure lah, tapi in reality, alright, it is much longer and more convoluted. Okay, alright, and then they extend into the body cavity where they are surrounded by hemolymph. So now this Malpighian tubule, alright, actually is longer and more convoluted. Right, extend into the body cavity. Maksud dia lebih kompleks di structure dia. Alright, and this tubule is surrounded by hemolymph. Hemolymph tadi lah. Dia punya yang sama macam blood kita. Alright. So here you can see first, right? What happened here? The epithelial cells of the tubule, this tubule, epithelial cells of the tubule actively transport salts and uric acid into the tubule. Alright, into the tubule lumen from hemolymph, because these tubules, they are all surrounded by hemolymph. So hemolymph is similar to our blood. Maksudnya it carries substances including all of these waste product. Alright, so contoh dia adalah the transport salts and uric acid. So these epithelial cells, right, epithelial cells of the tubule will actively transport salts and uric acid into the tubule lumen, masuk dalam lumen ni, from the hemolymph. So sudah daripada hemolymph ni, hemolymph kat luar ni kan. So masukkan by active transport. Okay. Alright, and then so when salts goes in, so salts, uric acid goes in, then what follows after that? Okay, tak ada jawapan. Huh? Water lah. Bila salts kan, salts, uric acid, all right, goes into the tubule. Okay, this will cause, alright, kat luar jadi more hypotonic, inside becomes more hypotonic. So that water from outside, from the hemolymph, goes into the tubule. Right, by osmosis. Okay. And then next, so now water, right, plus salts and uric acid, then moves into the hindgut. So hindgut is here, right, hindgut. Actually hindgut consists of the intestine. Ni stomach dia tahu, ni parts of the digestive tract dia. Stomach, intestine, and this is rectum and the anus. Similar, right? Padahal ni insects ni tahu. So dia ada semua, stomach, intestine, alright, rectum and anus. So now first, right, salts and uric acid into the tubule, lumen of tubule, and this is followed by water by osmosis. So now water, salts and uric acid. Right, so now the fluid moves into the hindgut, which is the intestine. So dia pergilah mengalir ke sini. Intestine and also rectum. Where, alright, salts and water are reabsorbed into the hemolymph. Right, because salts and water, they are useful substances. Sama lah macam kita tadi, bila the ultrafiltration, the filter out, right, all small molecules, kita ambil balik, right, the useful substances. So same as in this insect. Alright, so salts and water will be reabsorbed into the hemolymph. Ambil balik, masuk the hemolymph. Okay, whereas waste in this case uric acid, right? Uric acid. So waste are excreted with feces through the anus. So uric acid plus, right, ada dia punya waste product and then will be excreted here, right, through the anus. Faham? Faham ke? Do you have any question? No question. Okay, bagus. Alright, this is more. Tapi nanti baca sendiri. Alright, and this one also. This is the detailed explanation. Alright, of the same process. Nanti pun baca sendiri. right? Because the basic concept cikgu dah cerita. Okay. So you can also refer to your ABI study guide. Sama Okay, done with that. Okay. Next. And you nak break sekejap ke? Break dulu lah. Kita ada 2 lagi application. So we take 5 minutes break. After that we can deal with 2 more applications. After that we are done with this subtopic. Okay?
Baik, teacher.
Alright. Okay, semua orang dah ada. Dah, semua dah ready. Alright, dah ready, kita sambung. So let's continue. Two more application. Alright, so the first is consequences of dehydration and overhydration. Alright, consequences of dehydration and overhydration. Alright, so what is dehydration? Hydration tu apa? Body fluid
No water.
become like insufficient water, right? Water level. Okay. Whereas overhydration, terlebih lah pula airnya. Right, overhydration. Right,
Too
too much
much
water. Yeah. Alright, so dehydration is due to loss of water from the body. So body fluids become hypertonic. Alright, hypertonic. Why hypertonic? Loss of water. Right, loss of water from the body, right? So that body fluids become hypertonic. This is dehydration. Whereas overhydration, right? Overhydration is less common, right? Usually people akan alami this problem. Dehydration is common, all right, but overhydration is less common, right? And occurs when there is an overconsumption of water. Betul tak? So overconsumption ni jarang lah. Sebab minum air banyak-banyak kan kembung. Dan kita stop lah kan. Right, that's why this is less common. Right, so overhydration is less common, right? And occurs when there is an overconsumption of water. But dehydration is very common. Nah, saya rasa, I think hari-hari pun you all mungkin dehydration. Right, especially you all lah. So you all kan tak suka minum air kosong. Tak? Kan, nak minum mesti nak air yang berperisa. Okay? So most probably, right, there will be many. Okay, having this problem. So these actually the consequences of dehydration. Thirst, right? Small quantities of drug-colored urine. All right. So if you notice, your urine, right, is in small quantity and dark-colored. That tells you that you actually having this dehydration problem. Right, lack of water in your body. So tengok sendirilah, kan? Air kencing sendiri macam mana? So kalau sikit sangat tu every time urinate sikit saja, right? And very concentrated, dark coloured. So that tells you, right? You have you are being dehydrated. All right, and then lethargy. So lethargy, right, or severe drowsiness. So this is another consequence. All right, severe drowsiness. Okay. All right, exposure to higher levels of metabolic waste, reduced muscle efficiency. So this is what it means by lethargy, severe drowsiness. Okay, all right. Exposure, why? Because happen in exposure, the person is being exposed to higher levels of metabolic waste because due to water level that is low in their body, right? Because actually the importance of water too, actually it flush away, right, toxic or metabolic waste from our body. So kalau sikit maksudnya we will be exposed to higher levels of metabolic waste, right, and this reduce muscle efficiency as well. Okay, all right, and then next is low blood pressure, right? Low blood pressure, why? Due to reduced blood volume, right? Because lack of water, reducing blood volume resulting in low blood pressure. All right, and then next is raised heart rate. Raised heart rate due to low blood pressure. I mean, our heart need to work harder, right, by increasing the heart rate. Okay, so raised heart rate, low blood pressure, raised heart rate. All right, and then next is inability to lower body temperature. Why? Because lack of sweat. because no water to be removed from your body. Tak boleh nak sweat. Alright, because we know that sweating carries away heat from our body. Alright, so therefore lowering the body temperature. But due to dehydration, then we cannot produce sweat, so that more heat cannot leave our body. Alright, so having difficulty to lower body temperature. And in severe cases, seizures, right, brain damage and death. Okay, so it can be fatal also. So be very careful. So look out, right, at the amount of water that you consume every day. So this is dehydration, and then for overhydration, right, this is less common, occurs when there is an overconsumption of water. All right, so overconsumption of water will of course results in large volume and clear urine. All right, large volume diluted clear urine. Okay, maksudnya kita minum banyak air lah tu. And then swelling of cells due to osmosis, right, from hypotonic body fluid. Okay, maksudnya cells, right, because there's a lot of water in the blood, and then the water leaves, right, the blood, and then goes into our cells, causing, right, cells to swell. Okay, and then headache, disruption of nerve function due to swell cells, so including the nerve cells, right? This will cause interruption or disruption to nerve function, causing headache. All right, and in more serious cases, delirium, right? So an acutely disturbed state of mind, blurred vision, seizures, coma, and death. Right, so it can be fatal also, right, for overhydration. Okay, tapi jarang lah kan, macam saya cakap tadi lah, biasanya orang tak minum terlebih sangat. Okay, so ini saja, dehydration, this more, right, so you can add more lah. Besides these consequences, then these are other consequences. Alright, dehydration cause sleepiness. Ada tak? Samalah drowsiness tadi kan, siapa dalam kelas selalu ngantuk tu, check lah your water consumption every day, cukup ke tak? Alright, and then constipation, so those having constipation problem, also check again, right, your amount of water consumed. And then dry mouth or skin, this is another indicator. Alright, and dizziness, headache for dehydration. Overhydration, change in behavior, confusion, because tadi ada interruption to the nerve function. Alright, blurred vision, muscle cramps, nausea, and vomiting. Alright, so these are all the consequences. Okay, done lah. So tak adalah ni, because this one like state only. Okay, and then next is the last part. Alright, the last application, and the last part in this subtopic. Right, treatment of kidney failure by hemodialysis or kidney transplant. So actually the main thing here is treatment of kidney failure. So treatment of kidney failure, right, there are two. One is hemodialysis, another one is kidney transplant. But of course, kidney transplant you need to wait. Alright, need to wait for suitable kidney, ada donor, right? Then only it can be carried out. So maksudnya more practical adalah hemodialysis lah. Alright, so treatment of kidney failure focuses on two main approaches. Alright, so one is hemodialysis and another is kidney transplants. So it is a condition, right, kidney failure. So it is a condition in which the kidney fail to adequately filter waste products from the blood. So meaning that our kidney will no longer able to clean our blood. All right, because they cannot adequately filter waste products from the blood. It can be caused by injury or disease symptoms, vary depending on the seriousness and progression of the disease. All right, and then if not treated, kidney failure leads to death. Because never mind, because kidney filter out toxin from the body. So kidney failure will result in toxin to accumulates in our body. All right, so that's why they will lead to death. Okay, so this is about hemodialysis, right? Hemodialysis. So apa dia buat kat sini in hemodialysis? Cikgu besarkan nampak quite small here. Oh, aku tak boleh. Alright, so this is for hemodialysis. Alright, commonly called kidney dialysis. So it is a process of purifying the blood of a person. Kenapa lah duduk kat sini ni? Alright, so it is the process of purifying the blood of a person whose kidneys are not working normally. Alright, because tadi failure of the kidney to filter out, alright, to adequately filter out metabolic waste. So meaning that it is require help. So dibuat apa? Right, to clean the blood, then the person must go through this treatment, right, known as kidney dialysis or hemodialysis. So during hemodialysis, what happens is, right, the blood will be purified. All right, and then so this hemodialysis treatment lasts about 4 hours. And they have to spend, right, about 4 hours, and it is done 3 times per week. This one actually depending on the level of the kidney failure tu, right, how severe it is. But I think the maximum is this one, right, 3 times per week, 4 hours per session. Each session 4 hours, all right, and in a week kena go through 3 sessions. All right, so a person can be treated this way for years. Yes, cikgu pun dah dengar lah, ada orang dah bertahun-tahun they have to undergo this treatment. All right, so the concept here, they use fresh dialysate. All right, ni kidney dialysis lah. So you can imagine how this is huge, you know. You all pernah tengok tak this dialysis punya machine ni?
Pernah.
Pernah. How big it is, Arif?
Peti ais. Saiz peti
Peti
ais.
ais. Peti ais yang small tu kan? Yang satu one
Ada
door
yang small, ada yang
refrigerator,
besar.
right? One door refrigerator.
Ada yang kecil, ada yang besar. Be around that area lah.
Ada yang besar. Okay, alright. So you can see, right? Actually, that huge machine is to do the function of our kidney, right? To do the function of our kidney. Yang size dia macam mana? What's the size of our kidney?
Saiz penumbuk.
Ya ke? You ada search lah. Kidney size penumbuk, are you sure? Itu heart kita. Tengok kidney size. Cari-cari sekarang. What's the size of the kidney? Besar mana dia?
12 cm, teacher.
Huh?
Betul lah, teacher. The size of a fish or 10 to 12 cm.
Oh, ya ke? Oh, kidney pun sama Cikgu ingatkan size kita punya heart saja, size of our fist. Yeah, kidney size. Betul lah? Cikgu pun lupa sebenarnya. Kidney size normal. Yeah, 10 to 12 cm, about 5 inches. Alright, so about 5 inches, 10 to 12 cm. Alright, so that's the size of our kidney. Bila kita nak buat dia, alright, it has to be this huge. Alright, but this still is not as effective as our kidney. Okay, so nampak tak how complex it is kan? Kalau manusia dah nak buat tu memang susah lah. Alright, so ni kena reflect balik lah kan, kekuasaan Allah Ta'ala. Kidney yang kecil tu boleh buat all functions. If let's say we want to like imitate, right, nak create a machine that can do the function of the kidney, it has to be this huge, and this is still, right, not as efficient as our kidney. Banyak kekurangan dia. Alright, so they use fresh dialysate. Alright, so fresh dialysate contains no urea. Alright, so this one actually in exchange for with our blood nanti. So fresh dialysate, they use fresh dialysate which contains no urea. All right. Why? Because to encourage diffusion from the blood into the dialysate. So ni konsep dia. Sebenarnya dia letakkan, dia supply fresh dialysate. Alright. And after that, blood, right? Blood from the patient, dia bawa keluar, right? Taking out blood from the patient. And then pump, right? The blood into this dialyzer. Alright, and then they use this dialysate, fresh dialysate, they supply it into this dialyzer. Alright, and they have membrane separating this, right, dialysate and also the blood of the patient. Lepas tu, right, they akan encourage of the exchange of substances. Alright, so meaning that when dialysate has no urea, patient's blood has a lot of urea, so that urea will move into the dialysate. You understand the concept? Faham tak? So dialysate ni no urea. But blood, because our intention tadi is to remove toxin, right? Such as urea from the blood. So that's why there's no urea in dialysate, so that, right, bila they ada together, separated by membrane, alright, separated by membrane, so that the urea from the blood will move to the dialysate. Sebab dialysate no urea. In the blood has high urea. Faham ke? Okay, so first they use this fresh dialysate. And then, so this fresh dialysate, right, contains no urea to encourage diffusion from the blood. And then glucose and other useful molecules at optimal concentrations. Okay, this is to minimize loss from the blood. Because kita tak naklah orang tu nanti, bila after undergo this dialysis, dia tak ada dah all of the useful substances like glucose and other like amino acid, everything, right? Okay, we don't want to lose all of these useful substances, so that dialysate tu kena ada about the same concentration, right, as the blood, so that this will minimize the movement, right, of glucose and other useful substances into the dialysate. And then next is high solute concentration, right, remove excess water. So dialysate ni will be high in osmolarity lah, dia ada high solute concentration. Why? So that this will allow, right, for water to be removed from the blood, excess water to be removed from the blood. Alright, and then this dialyzer, the dialyzer. So dialyzer contains a semi-permeable membrane, alright, that allows small particles such as urea to diffuse through. But larger molecules and cells remain in the blood. So macam the ultrafiltration process lah. Okay. Alright. So this dialyzer, right? It has this semi-permeable membrane, right? That allows small particles to diffuse through. So meaning that urea boleh diffuse through this semi-permeable membrane, masuk dalam dialysate. Alright. Maksudnya urea from the blood, sebab tadi ada high urea content kan? Okay, so bila pump into this dialyzer with the presence of semi-permeable membrane, urea, alright, urea will diffuse through the semi-permeable membrane right into the dialysate. But then large molecules, right, and cells remain in the blood. So preventing other large molecules passing through, so that they remain lah dalam blood of the patient tu. Maksudnya berjaya tak dia nak remove urea tadi? Berjaya lah. Faham tak? Faham ke so far?
Teacher.
Faham. Okay. Alright. Okay. And then next is use dialysate then. Right. Use dialysate. So meaning after, right, exchange of materials happens here. Yeah, water will be excess water, right, will be removed, and then urea also removed, right, but then useful substances remain in the patient's blood, and also their blood cells, everything, larger molecules all remain inside of their blood. All right, and use dialysate, meaning that this dialysate now collects, right, all of the waste, including the urea. So use dialysate collects filtered out small molecules such as urea. Okay. So yang filtered out tadi through the semi-permeable membrane will be collected by this dialysate. Meaning that use dialysate will have high content of urea. Okay, high urea level and probably water level as well. If, right, the blood of the patient has excess water. Okay, all right. And then saline solution here, right, given actually prevents excessive water loss, which could lead to dehydration. Maksudnya dia adalah method of monitoring also. All right, to prevent excessive water loss from the patient. All right. Okay, so inilah dia. So this is the concept. All right, next. So other molecules and ions are present, right, and therefore filtered or balanced. But these are the key components of the dialysate. All right, maksudnya in preparing this the property, right, or the components of fresh dialysate is very important actually to make sure, right, the blood can be cleaned. Okay, maksudnya mencapai objektif lah. Maksudnya if let's say we want to remove urea, then dialysate mesti tak ada urea. So that, right, urea will be excreted out, will be removed from the blood. Alright, and then in order to remove water, excess water from the blood, maksudnya dialysate kena ada high solute concentration so that it is hypertonic to the blood, right? Therefore allowing water, excess water from the blood to be removed, masuk dalam dialysate. Alright, however, like glucose and other useful substances, we don't want, right, the patient to lose all of this useful. Maksudnya dialysate kena ada about the same optimal concentrations, right, with the blood, so that this will minimize, right, movement of these useful substances from the blood into the dialysate. So that's the concept. Faham? So bila semua dah remove tu, and then they supply balik, right? Supply balik the clean blood back into the patient, masuk balik clean blood. Alright? So nampak tak? So this actually dialysis machine, they imitate the structure, the function of the kidney. Faham ya?
Faham cikgu?
Okay, good. Wait, macam mana lah cikgu nak buat benda lah ni? Okay, wait. Cikgu nak gerakkan. Okay, this is cikgu tambah saja ni so that you can make comparison. Alright, dialysis fluid composition versus the blood plasma composition. Alright, so nampaklah the difference. You can see level of blood urea. Where's urea here? Blood urea. Dialysate kosong. Urea tak ada.
Dia vary kot teacher, urea.
Apa dia?
Dia tak tulis sebab depends on people ke urea tu
Ya,
banyak
betul.
mana.
Ya, betul. Actually, di dialysate memang actually dia buat depending on patient. Kan cari source lain ni. Cikgu pun tak sempat jaga dengan tapi macam ada urea, tak ada pula. Tak apa, nanti cari lain lah. You all pun boleh cari sendiri tengok. Ni cikgu ubah lah ni. Alright, this is more on the dialysate composition. Tapi baca lagi lah. Dia tak adalah tanya detail Cumanya the basic. But the basic, right, the difference between dialysate and also blood tu kena ada. So the main thing you must know about urea. Alright, high hypertonic, right, or high solute concentration in fresh dialysate, and glucose and other useful mesti at optimum concentration. So this one you must know. Okay, alright. Yang additionally tak tahu pun tak apa, tapi nak tahu nanti cari sendiri. Alright. So sama lah ni, it's the process. Alright, and this one all is everything that I put in here slides. They are all additional information. Okay, so this one is about how, right, the blood actually taken out. So leaving the patient through this, they call it as arteriovenous fistula, this part. Actually the combined vein and artery. Okay, so blood goes out from this arteriovenous fistula. All right, and then going through this dialyzer. All right, dialyzer, so that this you can see the artificial membrane. Right, semi-permeable membrane. This is dialysate, right, fresh dialysate used, and this is the blood of the patient, right, so that all of the toxins and excess water be removed from the patient's blood. Alright, other useful substances remain. After that, after the blood is clean, and then dia akan return to the patient back to this AV, arteriovenous fistula. Okay, ni cikgu tambahlah what is AV fistula. Right, AV fistula, so nama panjang dia tadilah arteriovenous fistula. Right, simplified as AV fistula. So where they connect, right, an artery to a vein, usually in the arm, to create an AV fistula. All right. So an artery is a blood vessel that carries blood away from the heart. A vein is blood vessel that carries blood back towards the heart. All right. So when they connect an artery to a vein, the vein grows wider and thicker. Alright, so jadi makin wider and thicker. They combine characteristic tadi kan. So making it easier to place the needles for dialysis. So the AV fistula also has a large diameter, right, that allows your blood to flow out and back into your body quickly. Okay, alright, and then the goal is to allow high blood flow so that the largest amount of blood can pass through the dialyzer. Alright, ni pun additional lah ni. So that you can better understand lah. Alright, and then next is, so dan kita discuss tadi is hemodialysis. Hemodialysis. Alright, so next is kidney transplant. So this is about kidney transplant. So you can see, right, a grafted or transplanted kidney put here, placed here. This is how actually they carry it out, like kidney transplant. So a transplant is the best long-term treatment, of course lah kan. So you can replace your kidney with a healthy ones. So this is actually the best long-term treatment. Alright, so donors can be either someone who has recently died. Nah, ni siapa yang nak derma organ tu kan. Tapi derma organ ni, what do you think? Is it allowed in Islam actually? Organ, to donate organ?
It is, teacher. It's allowed.
It is allowed.
It's allowed with a few syarat, tapi it's allowed.
Yeah, actually it depends on different view, right? Because some ulama says it is allowed, but some ulama says it is not. Cikgu pun dulu, right, last time, cikgu memang believe that from my reading lah. Cikgu tengok, okay, most of the sources says that it is allowed. But recently I heard, baru je, I think sometimes this year, I heard like one ustaz saying that to him it is not allowed, and then there are the support, there are the like supporting material from ulama yang dulu-dulu, but I can't remember. Alright, which says that sebenarnya ianya tidak dibenarkan. Okay. So wallahu alam lah, kita pun kurang ilmu kan, tapi boleh tanya lah kot orang lebih baik dan cari juga sources, right, daripada ulama-ulama yang terdahulu. Okay, so donors, right, donors can be either someone who has recently died, alright, or a person who has chosen to give up one of their 2 kidneys, right, because we can survive with only 1 kidney. Okay, so kalau ada orang nak derma, boleh lah. Alright, and then donors and the recipient have to be a close match, alright, in both blood and tissues. Okay, this is to minimize the chance of rejection. Remember, our immune system will identify whether the cells actually belongs to us or not, right? So kalau tak sama, tak match, maka our immune system will reject it, will actually destroy, because they will treat, right, the kidney, transplanted kidney, as foreign. Therefore, right, this will trigger antibody production, which will destroy, all right, the transplanted kidney. Okay, right. If let's say semua ni dah fulfill, meaning that there's no rejection, so the transplanted kidney is grafted in the at the lower, right, abdomen with the renal artery, renal vein and ureter connected to the recipient's blood vessels and bladder. So dia sambung balik lah, right, all the renal artery, renal vein and also ureter, semua dia connect balik to replace dia the Dia fill kidney itulah. Okay. Right. Itu saja. And then this one, ada lagi. Masya Allah. Cikgu kan dah 2 application saja. Rupanya ada 3. Maaf lah ya. Boleh ke? Habis kan? Boleh? Penat dah?
Boleh.
Kalau satu je lagi. Takkanlah kita nak biarkan satu ni kan? Alright, maaflah cikgu dah beri janji palsu tadi tu Saya ingat boleh dah spend satu period for you to do your revision. Rupanya janji palsu, Masya Allah. So cikgu minta maaf ya. Rupanya tak sempat nak beri masa itu. Tak apalah, probably nanti test kita start lewat sikit. Right? Kita start test lewat sikit lah. Nanti cikgu tengoklah aku apa. Okay Alright, so the last one, last one is blood cells, glucose, proteins and drugs are detected in urinary tests. Okay, so blood cells, glucose, proteins and drugs are detected in urinary tests. So this is called as urinalysis, right? Analysis of urine. So diagnostic tests of urine samples provide an easy, effective way to detect illness and also drug abuse. Alright, so it is a clinical procedure that examine urine for any deviation from normal composition. Maksudnya kalau normal urine sepatutnya macam ni. Right, ini ada, ni tak ada. So when doing this urinalysis, right, they can tell if let's say the person is taking drugs or not, right, or having any disease or not. Alright. Okay, so processes that affect the metabolism affect the metabolic waste, right? That is produced and then excreted in urine, because urine actually contains all of the metabolic waste. So kalau metabolic process kita semua normal, maka the urine punya component will be normal lah, alright. But if let's say something is wrong, right, in our body, then that akan affect metabolic waste produced, and this can be detected right in urine. Okay, so example, presence of blood cells. Sepatutnya blood cell boleh present tak dalam urine? Boleh tak
No.
blood cells present? Tak boleh. Sebab dia tak boleh lepaskan di ultrafiltration process. They cannot pass through the basement membrane, right? And also the podocytes. So there shouldn't be any blood cells in our urine. Okay, so meaning that if let's say blood cell is present, it actually indicates infections, right? Disease and some cancers. So ada soalan dia tanya, if blood cell is detected, right, in the urine of a person. So the question asks, which part of the kidney that is being damaged? Kat mana yang rosak? Dekat mana?
Glomerulus.
Yes, glomerulus. Right, glomerulus and Bowman capsule probably lah. Tapi I think the best one will be glomerulus. Alright, and then glucose is a strong indication of diabetes. Okay, so supposedly, because last time when we learned about selective reabsorption process at the proximal convoluted tubule, 100% glucose will be reabsorbed. Is that right? Betul tak? So if let's say glucose can be detected in our urine, therefore it indicates very high glucose level in the blood until kita gagal untuk reabsorb semua. Okay, so this indicates diabetes. If let's say glucose is detected, right, in our urine. Okay, and then trace amounts of proteins are normal, right, because some proteins are very small, but larger amounts indicate kidney disease. Protein pun tak, tapi some proteins are very small so that they can pass through, alright, during the ultrafiltration process. But the larger amounts of protein, right, in urine indicate something is wrong with the kidney as well. Sama lah macam ni ultrafiltration lah glomerulus. Alright, and then drugs. Drugs or their breakdown products can often be detected in urine samples, right? Because drug is small enough, right? So they can pass through the ultrafiltration process and then can be detected in urine sample. Okay.
Teacher,
Yes.
teacher yang dia cakap some proteins amount, apa tu? Some proteins, yang dia cakap ada trace amount of protein
Trace
tu.
amount of proteins.
Meaning like kalau in urinalysis tu, dia detected jugalah.
Yeah. They can compare the
Hmm.
amount to present. Banyak ke sikit? Kalau sikit, that one is normal. Tapi kalau banyak, that indicates something is wrong. Okay. Alright. And then, so this one ni ada lah ni, tapi cikgu pun tak reti lah nak baca macam mana. Right. This one actually the test and reading time. Okay, so this one is composition, right, of urine in healthy person. So basically this one lah, maksud reference dia boleh guna kat sini lah. So kalau dia less ke more ke, then that indicates something is wrong. Okay, alright. So boleh tengok nanti. And then this one pun dah additional lah. Ni cikgu dah cerita dah before. Alhamdulillah dah habis. Alhamdulillah finally. Okay, do you have any question? Ada soalan tak so far? We have finished 11.3. Right, we have covered all. Ada soalan? Tak ada. Tak ada soalan, tak apa. Cikgu, awak pun dah penat kan? You pun penat. Okay. So petang ni nak start kau apa? Setengah, 3:20, 4:20. 3:15? Tak boleh?
Boleh
3:20?
teacher.
Apa dah?
Saya rasa boleh ke teacher.
3:15 kan? Alright. So then we start our test at 3, tak apalah, cikgu beri 3:20 lah, 5 minutes lagi. Right, so 3:20. Maksudnya kita cikgu kena inform dia ada kelas lah. Right, so 3:20. Our test start at 3:20. Okay, right. So before that, maksud you nak buat revision sendiri kan? Tapi join Zoom earlier lah, because start nanti 3:20. So 3:10 tu join Zoom dah lah. Tapi nak buat study, nak buat revision sendiri, go ahead lah, no problem. Cumanya join Zoom dulu around 3:10. Boleh? Right, because you nak tengok attendance semua ada ke tidak tu. Nanti takes time pula. Okay ya? We will start sharp at 3:20. The test start sharp at 3:20. So Zoom, cikgu share nanti dalam 3:10. Okay ya? Okay, so I think that's all. So cikgu pun tak wrap up. Alright, pun dah penat. So nantilah kita wrap up. Alright, cumanya cikgu cuti. Cikgu ambil cuti. Alright, dari hari esok sampai Jumaat. Sampai Ahad lah maksudnya. Alright, so tomorrow kalau ada kelas, nanti cikgu tak akan, I cannot remember my timetable, you know, because keep on changing kan. Nanti cikgu berilah, tapi maksudnya there's no like online class macam ni. So you all nak buat apa?
Esok tak ada teacher. Yang ada bio Khamis memang cuti. So Friday je. First period.
Okay lah, buat Kahoot lah. Boleh buat Kahoot. So maksudnya buat so that time because we have finished everything, so go through right 11.3 on your own ataupun ada discussion with your friends pun boleh, in your RP group pun okay. Alright, and then buat Kahoot. Okay. Boleh? So maksud next week baru kita start dengan 6.6 part 2 and followed by the last one 11.4. Okay. Boleh ya?
Boleh teacher.
Alright. Okay. Okay, so all the best. I wish you all the best for your test. Moga-moga more 7 this time. InsyaAllah. Amin. Alright. Okay, so I guess